Hi Hires, so we are now looking at Key Area 2, Plant and Animal Breeding in Sustainability and Interdependence, and we're looking at plant field trials. Okay, so the whole point of a field trial is to test a particular cultivar against different conditions, or test different conditions on their own. Okay, so field trials are set up exactly as this picture says, in a field with sections of that field that are going to be applied with different things. So loads of reasons why you might want to run a field trial. So you could look at different crops, you could compare one crop under different environmental conditions, you could test a particular treatment, so that might be a pesticide, that might be a fertilizer, that might be a combination of different things, and you might be looking at evaluating a new cultivar that you're looking at, and that could be through breeding or through GM crops. So none of this has changed from, frankly, any time you've ever spoken about a science experiment. You have to make it fair. Okay, so to make it fair, what you have to do is say that you know for certain that the independent variables, that is the thing that you changed, is the factor that affected the dependent variable, which was the thing that you measured. Now, you genuinely have to be very, very clear on what is the difference between independent and dependent, because that's how they're going to use those terms now, no longer just saying what was measured. Okay, so make sure you're happy with those terms because they come up again and again and again. And yeah, from, as I say, early doors, this means you can only change one thing. So the things that you need to be able to talk about in terms of specifically plant field trials is how you choose your independent variable, replication, because you always need to repeat for reliability, and one that we haven't really spoken about a lot, in fact I can't think that I've spoken to you about at all, so avoiding bias. So our selection of treatments, that's kind of obvious. Okay, so have you got the same size plots? Have you set them up so they are the same in terms of how many plants you put in? Have you only changed one thing in your basic setup? Okay, so you could be looking at things like type of or amount of fertilizer, types of pesticides, amount of pesticides, that kind of thing. And so these are carefully set out systems. Replication is kind of obvious. You should recognise that any sample will have variation. Okay, the less variation that you have in your results, the more you trust those results, because you're saying that's within a very, the narrower and narrower the range. But you're looking at living things, so you expect variation. So you're not going to take just one sample, and you're not going to have just one plant. You're going to have many plants within that sample, and then you're going to have external kind of replication of that sample set as well. So repeating for reliability. A lot of replicates normally, 3 is your minimum. We normally say that for doing any kind of test or experiment in class. We would say you want a minimum of 3, but genuinely in most experimental stuff, if you want to prove that you have something that is working well, you would expect a lot more. And especially, and this is really important, that you would make a decision about how many replicates you wanted to have for you truly to be able to say this is a reliable set of data, is if your first set of results gave you a wide range. Right, so avoiding bias. So to avoid bias, what we're talking about here is if you don't set up your experiment well, then you can end up with you already slanting the results, pushing them in one direction or another. That's what we mean by the bias. So, right, have a look at this plot. Okay, so these are obviously neatly set out plots. That's fine. They all look alike. That's also fine. That's what you want. Okay, right, now imagine this was different fertilizer levels. Okay, so in this field you have 5 rows each level. That's good. That's good replication. You're going to have hopefully sections of data for each one of those. That's all good. Right, now imagine at the end of this particular field we have down here a stream. Okay, so what we have now is water at a higher level at this end of the photo, and as we come this way we get less and less. Now that means that you have got a problem. So say your high fertilizer was at this end and your low was at that end, that's not the best colour, but never mind. Okay, then this high fertiliser is actually testing high fertiliser and low soil moisture, in comparison to this one, which is actually testing low fertiliser and high soil moisture. So we've got a problem. We've introduced a bias into this data set because we've effectively made it two experiments. So what you need to do is to randomize your plots. Okay, so what we're looking at in this one, we have here. Okay, so what we have here is an example of a randomized plot set. So at 3 different locations, okay, we have 2 different treatments, so the kind of grey squares and the white squares. And so this means across this you have 90 for each treatment. And what you've done is you've just put a random spots and dots as to which ones to go where. No bias. Okay, so this term bias you need to be able to use, and you need to talk about avoiding bias by using this term, randomization. Okay? The last thing you have to take into account is that you want to, in fact, I already mentioned it really with the bias one, you need to try and do a range of environments, because otherwise it is difficult for someone to defend the results, because you might have someone saying, well, no, but just because of where that particular field was that you did all those trials in, it's a particularly sunny spot, and if I put that in my farm, it's not going to make a difference. Okay, so what you need to do is to test it, but to test it in different environmental ranges. And that's really just to get round all of the factors that would normally impact in photosynthesis. So temperature, your nutrient availability, your water levels, and light levels. Okay, and that's us.